Yes, welcome back to the Pioneer Room. A continuation on The Biology of Belief by Bruce Lipton. Yeah, doctor that is. Now, I am happy that you are sharing content like this because Bruce Lipton is from the scientific fraternity. So I can speak on peer-reviewed scientists, alright? And if he is releasing content like this, then I can take it to the next level. Thank you. Now, oftentimes I come here, I talk about epigenetics. I don't know how many people understand it. You might Google and still don't understand. But let me break it down real simple so that even the baby can understand, right? Now, epi means above, right? And genetics is the study of the genes, right? So we're looking above the genes. That's epigenetics. But let me go a little bit more in depth by describing what is called the omics technology. All right. In science, there's what is called omics technology or omic platforms. All right. This is what it's made up of. For example, there is a tumor in the brain, all right? This is how the technology is applied, and I will use it to explain epigenetics and the biology of belief. There's a tumor in the brain. This platform allows you for targeting the gene of that tumor. So targeting the gene associated or correlated with the tumor, studying that gene is called genomics. So the first piece of the omics platform is genomics, studying a targeted gene. All right, this in this case is the tumor in the brain. Now the next piece to the platform is studying the epigenes that turns on that tumor gene. Okay? So the study of these epigenes or these conditions or these factors is called epigenetics, epigenomics. Some might say epigenetics, but epigenomics or epigenetics, fine. Okay? Then a condition, switches on or switches off a particular gene. We said the condition is the epigenes. Okay? Now, what is responsible for reading the genes when it was switched on or switched off are species they called mRNA. The process of studying these mRNAs that can read the DNA. When the gene is switched on, it reads the DNA. If the gene is switched off, it can read the DNA. Okay? The study of these mRNA is called transcriptomics. So we've gone through genomics, we've gone through epigenomics or epigenetics, and we are now at transcriptomics. mRNA reading the DNA. Now, after mRNA reading the DNA, the next step of the process is where the mRNA is transcribed into protein. So we have proteinomics. So we have genomics, epigenomics, transcriptomics, and now we have proteinomics, where we're studying the proteins now from the genes. Okay? Proteinomics. Following? And then, when you arrive at the proteins give rise to metabolic functions in the body or in the brain. The study of these metabolic functions is called metabolomics. Okay? And the metabolomics is because they are studying the metabolites, which give rise to your physiological processes. Physiology. So your physiology is as a result of your metabolites, which comes from your biochemistry. So the study of your biochemistry and the metabolites derived is metabolomics. Now, metabolites control your physiology or your physiological processes. You now gone to the doctor, and the doctor will tell you have high blood pressure, low blood pressure, diabetes, arthritis, and all these other things, right? In summary, the platform, the omic platform, is comprised of genomics, epigenomics, transcriptomics, proteinomics, metabolomics. And this is how they study organisms in this cassetted form. And this is how you understand genes. Now, the study of genomics, epigenomics, transcriptomics, proteinomics, metabolomics is for a physiological outcome is what you call epigenetics. What is the pathway? When you turn on a gene, what is the communication crosstalks at the epigenetic level, at the mRNA level, at all the protein level? What is the crosstalk? What is the communication? And every gene that is switched on follow a unique pathway, epigenetic pathway that must be preserved, maintained. This is why you have genes inside the body called conserved genes. Nothing can move them or change the pathway over millions and millions and millions of years. They are called conserved genes. You can Google them. The epigenetic pathway in these are wired. This is where the science is at, trying to alter these pathways. Okay? But there are other genes that are not conserved, right? And environmental conditions can change them. This is where you have the genomic section and the epigenomic section. So in between genomics and epigenomics, what can you use to change the epigenomics? This is where the science is at. Gene editing. This is where you apply things like the CRISPR tool. This is where you can ground yourself and modify the epigenetic outcomes. This is where the sun is important. This is where astrocytes are important. This is where grounding, electron, photons are important. This is where you alter the frequency for the epigenomic outcome. This is the most important section of the omics technology. This is why I'm saying, giving you the ability to hijack your brain, this is the section in which you do it. You hijack it at this, between the genomics and the epigenomic stage. Using various techniques, meditation, yoga, foods, anything, any signaling you can use to, or even thoughts, you can use to change the epigenomics. Once you change the epigenomics, everything after that will change to your desired outcome. All right? So this is what is epigenetics. Thank you very much.